Dear students, welcome once again to the online classes of biology. So, children, we are again in a situation of pandemic and the COVID cases are increasing day by day, and it is the time as biology students you should be aware how to prevent this, isn't it? You know that how to prevent, but because of our negligence, okay, all the problems comes. We can prevent this by wearing mask properly, isn't it? Only the means even vaccination sometimes cannot prevent what the disease. So the only way to get escape from this is what wearing the mask properly I said, isn't it? Because wearing mask everyone is doing, but not wearing properly sometimes, isn't it? And what keeping distance and sanitizing your hands or washing your hands before you touch your face. So this all, as it is a kind of airborne disease, we have already studied, isn't it? Diseases chapter, how they spread. Okay, so airborne diseases can enter only through the what nose or through the mouth, etc., isn't it? Means especially through what breathing, inhalation. So you have to cover your nose properly and before touching your hands, as what you that can also spread by hands. So frequently wash your hands if you are going outside. Okay, keep distance, try to keep distance. Okay, and don't go to crowd, and all this are knowing, everyone is knowing all this, but we are not following it strictly, isn't it? So as you heard, prevention is better than cure. So we will try our best to prevent ourselves. If it comes, then we will see, isn't it? Cure is also that there, but prevention is the first measure that we have to do. And as biology students or social means as the members of the society, it is our duty to, means if one means one can spread the disease to many others. If we are able to prevent, okay, the disease coming to us, we can prevent the disease spreading from us to many others, isn't it? So it's our duty, and to means take all these precautions. No need of seeing others. What we have to do, we have to do that. Understood? What we can do, we have to do that, isn't it? And it's our responsibility as the members of the society, okay, to prevent the occurrence of disease as far as possible. What we can do, we have to do for this purpose, isn't it? And again, online classes are going to start, and it is not lockdown for your studies, it is only lockdown for your offline classes. So continue your studies, okay, study properly, and get knowledge also, not only for the examination. You read the textbooks and all other things, so that you will get more knowledge also. Okay, so utilize the time. Instead of wasting your time, you can read more and more. Take the opportunity to read and acquire knowledge. Okay, so let's continue the chapter, that is biotechnology, principles and processes. I have already explained you about what is biotechnology, isn't it? That is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. I have explained you the simple meaning of this, isn't it? This is the definition, okay, given by EFP. So what European Federation of Biotechnology's definition it is. And I have told you, if any living systems, microorganisms, okay, if they are used for the products or services of human being, then it can be considered as what? Biotechnology. And I have also explained you what is genetic engineering and how we can maintain, okay, or what is the importance of maintenance of sterile, okay, conditions for the what success of the biotechnological products or biotechnological procedure, isn't it? And we have also seen what is recombinant DNA, isn't it? And then what the technique of genetic engineering includes what? Creation of recombinant DNA, use of gene cloning and gene transfer, and this is done with the help of various tools. So we will have to study about the tools today. Okay, also I have explained you the first the recombined DNA molecule, isn't it? The construction of the first recombinant DNA, that also we have studied in the offline classes. So let's study the tools that are needed for the recombinant DNA technology. What is recombinant DNA? DNA means containing any other foreign genes, and what means recombination is done in the genome, that is called as what? Recombinant DNA. So for doing this we need some tools. If you need to do any any practical or any surgery, you need some tools, isn't it? So what are the tools that are needed for the recombinant DNA technology? Okay, so the first one is the restriction enzymes. Okay, so there are restriction enzymes, polymerase enzymes, ligases, vectors, host organism. All these are the tools, okay, with which we can accomplish the recombinant DNA production. Understood? So the first one we are going to study is restriction enzymes. So restriction enzymes, how we can understand. Okay, so in the year 1963 there are two enzymes, that is, you would have heard about bacteriophages, which attack bacteria, E. coli. Okay, so two enzymes which restrict the growth of bacteriophages in E. coli, means which prevent the growth of bacteriophages in E. coli, were isolated, and in two different ways they were working. One was cutting the DNA, okay, and the other one was just adding methyl group to DNA. So by adding methyl group to DNA or by cutting the DNA of bacteriophages, okay, they were, means the bacteriophages were not able to infect Escherichia coli or E. coli. You know, E. coli is a bacteria and bacteriophages are parasites, isn't it, that grows on E. coli. So due to this means two enzymes could prevent the growth of bacteriophages on E. coli in two different ways. One was cutting the DNA and the other is what? Adding methyl group. Understood? So the first restriction endonuclease, okay, that found was Hind2. Okay, so Hind2 was the first what? The restriction endonuclease that was found. Okay, and uh, and this Hind2 can cut the DNA at a specific site, okay, of 6 base pairs. Understood? And that particular specific means no other sequence, through no other sequence Hind2 can cut the DNA. Only there is one specific sequence containing 6 base pairs, and through that only Hind2 can what? Cut the DNA. And this was found after 5 years later, 1963. Okay, and it was found that this Hind2 can cut the DNA only through certain specific sequences. That sequences are known as palindromic sequences. I will explain that. Understood? And nowadays, after this Hind2 discovery, there are many other restriction endonucleases found. Okay, there are now around 900 restriction enzymes are there that are isolated from around 230 strains of bacteria. Okay, and each one will have specific recognition sequences. Hind2's recognition site will be different from EcoR1. Okay, so likewise, this 900 restriction enzymes, they will have 900 different recognition sequences. Okay, So this is about the naming of enzymes that I'll tell you later. So what are this restriction endonucleases or restriction enzymes? They are a group of enzymes coming under means a large class of enzyme called nucleases. Nucleases are what? Enzymes that can cut the DNA. We have learned this in the experiment, isn't it? In DNA's, DNA was found as the genetic material in that confirmation experiment was there, isn't it? Avery, MacLeod and all did. So in that it is clearly we have seen that they used nucleases, RNases and proteases. So nucleases are enzymes which can cut the DNA. Now nucleases can be of two types or they are of two types. What are they? Exonucleases and endonucleases. So exonucleases, they can cut the DNA from the ends. Okay, and endonucleases, they cut the DNA within the means specific positions within the DNA, means in between, not from the ends. So exonucleases and endonucleases, you understood now? These are nucleases only, they are nucleases enzymes only, but they act in two different ways. One can cut the DNA from the ends, which one? Exonucleases. And endonucleases in between, understood? Now this restriction endonucleases are a kind of endonucleases only. Okay, endonucleases, but the problem is they can cut the DNA through certain specific recognition sites called palindromic sequences. What are they? Palindromic sequences, understood? So palindromic sequences are what then? Listen, the word Malayalam, isn't it? Malayalam. You can read from here also Malayalam, and from here also you can read Malayalam, same. Okay, isn't it? From both the sides you can see that the word will be same if you read from both the sides. Madam, Madam. Okay, so from both the sides it's same, understood? So likewise palindromic sequences can be read same. Okay, so palindromic sequences are those sequences of base pairs that reads the same on two strands when the orientation is orientation of reading is kept same. For example, okay, 5 dash to 3 dash direction, if you read, okay, GAATTC, okay, so this will be same. It will be means it will be read same if you keep the orientation of reading same. 5 dash to 3 dash, if you read it from here also, it is same, understood? And you can see that they are complementary also, isn't it? G GAATTC, that is complementary to the other means nucleotides in the other strand, isn't it? G is combined with the C here, A T, isn't it? Likewise they are complementary to each other and they are in the two different strands, but if the orientation of reading is kept same, they can be what? Read same. So what are palindromic sequences that can be asked separately? So what are they? They are sequences of base pairs that read same on the two strands when orientation of reading is kept the same, means they are present in two different strands. They are complementary base pairs also, and they can be read the same on both the means on both the strands if the orientation of reading is kept the same, understood? So this is what palindromic sequences. So I told you each what the uh, restriction endonucleases can cut the DNA only through specific palindromic sequences. Okay, specific palindromic sequences are there. So we can see that one another what the end restriction endonuclease eco R1. Okay, so eco R1. So let's talk about the naming of this, how the name came, eco R1. So the convention for naming this enzyme, that is the first letter of the name should come from the genus. Okay, and the second two letters from the species of the prokaryotic cell from where it is getting. Okay, so eco R1, Escherichia is the what? The genus name. So from that E came, and Coli. Okay, so from that CO came, and R is coming from the name of the strain. Okay, so you know genus, species, strain, likewise it is there now. And then one came from the sequence, okay, or order in which the enzyme were isolated. So eco R1 means this is the first enzyme isolated from Escherichia coli strain. Understood? So like this only the naming of what the restriction endonuclease is done. Understood? So like that eco R1 can cut the DNA through the palindromic sequences, that is containing GAATTC. Okay, and you can see that this restriction endonucleases, they inspect the whole length of the DNA, and once they found that particular one, it will bind to the DNA and cut each of the two strands of the double helix, and that too at specific point. You can see here in this diagram. Okay, the restriction endonuclease eco R1 can cut the DNA wherever GAATTC is there. Understood? But they will cut in between the specific base pairs also, between G and A. Understood? Here also between G and A only it will cut. Okay, so this is the vector DNA and this is the foreign DNA. So what happens is, as they cut through the specific what the base sequences, you can see here this much will be removed from this strand, isn't it? And this part is just projecting out, and here where it cuts, here in between G and A. Okay, so what will happen, here this will become the free end, and here it will become free here. So they produce sticky ends. Understood? Sticky ends means, now you see this two are complementary, these two strands are having complementary base pairs. So once they come close to each other by hydrogen bonding, they can easily bind to each other. Understood? So this is the way, this diagram you have to learn steps in the formation of recombinant DNA by action of restriction endonuclease eco R1. Okay, so eco R1, first what they do, they inspect the whole length of DNA, and both in vector and in the foreign DNA. Foreign DNA we have to cut first, then we have to ligate it to the vector DNA, isn't it? Vector DNA molecule, like it can be plasmid or it can be a virus, whatever it is. So whatever we take as the vector, that also has to be cut, and the gene which we are going to take as the, means it is the, which is the gene of our interest, that also should be cut. So that both the cutting will be done with the same enzyme. Okay, so that they produce sticky ends. Understood? And when they brought together, they will bind by hydrogen bonds. So is it clear? So in this way only, what restriction enzymes produces what? Sticky ends. Okay, so this can be asked. Restriction enzymes cut the strand of DNA little away from the center of the palindromic sites, but between the same two bases on opposite strands. Okay, so that they produce what? Single stranded portions at the ends. Okay, and these overhanging stretches are called what? Sticky ends. Okay, so it can be asked how they produce the sticky ends, how restriction enzymes produces sticky ends. So what you'll write, you have to write that restriction nucleases cut the DNA little away from the center of the palindromic sites and between the same two bases on the opposite strands. Understood? And this causes what? Single stranded portions at the end, and these overhanging portions are called as sticky ends. They can easily get bonded by hydrogen bonds. Understood? So, this is how we are able to use what restriction endonucleases. Okay. As we are using the same restriction endonucleases to cut the DNA of the plasmid and the what the foreign means the host or foreign organism, same sticky ends will be there and easily they can get included. So this diagram is also important. Diagrammatic representation of recombinant DNA technology. Okay, so here you can see foreign DNA is okay, same restriction endonucleases, enzymes cutting both foreign DNA and the vector DNA at specific point. So what will happen? Sticky ends will be formed here, isn't it? Here also sticky end will be there, and in this part also sticky ends will be there, and that will be joined in the that can be joined together in the plasmid. Okay, ligase join foreign DNA into the plasmid, and then what can be done? The recombinant DNA molecule can be, that is, if it is a plasmid, it can be entered into the what host cell, and as the host cell divide, the gene of our interest also will get multiplied, amplified. Is it clear? So this is the way how it is done. So you understood now. The first is tool that is used for recombinant DNA means, what is recombinant DNA? DNA containing the gene from some other organism. Okay, so that is done with the help of what? Nucleases. Which type of nucleases? Restriction endonucleases. What are restriction endonucleases? It's a kind of endonucleases. What are endonucleases? They are nucleases that cut the DNA through, means in between, from inside. Understood? And restriction endonucleases can cut the DNA only through certain specific sequences. What are they known as? Palindromic sequences. So what are palindromic sequences? They are what palindromic sequences can be defined as the sequences of base pairs that read same on the two strands when orientation of reading is kept same. Okay, so what is the palindromic sequence for eco R1? From 5 dash GAATTC, and from where it will cut the DNA between G and A in both the strands, so that they produce sticky ends. Okay, so the sticky ends, same enzyme when we use for cutting the host cell or the foreign gene and the vector. Okay, they can come easily and join between them. Understood? So this is the restriction endonucleases and how they are useful in the process of biotechnology. Hope this much is very clear. Let's continue in the next class. Thank you.